Hello. I would like to thank the organizers of this meeting, especially Alexander, for inviting me. My topic is diversity and evolution of ion channels. And now I'm trying to imagine what did the ancestor of voltage-gated calcium and voltage-gated sodium channels look like. So voltage-gated sodium and calcium channels are members of large family of 4 repeat voltage-gated calcium channels. These calcium channels composed of 4 homologous repeats in a single polypeptide chain. There are 5 well-known and well-characterized subfamilies in this family. First of all, is voltage-gated sodium channels, but most of them are calcium channels indeed. LVL calcium channels, T-type, high voltage is low voltage activated, yeah? High voltage activated calcium channels is L, P, R, and so on. And these 2 subfamilies are not very relative to each other. Yeah, it's 2 different subfamilies. First subfamily is so-called sodium leak channels, voltage insensitive, but again, most of sodium leak channels, so-called, are calcium channels. Yeah, and these 4 subfamilies are almost exclusively belong to metazoa, to multicellular animals. And the 5th subfamily is voltage insensitive, and this is the store-operated calcium channels of fungi, CCH. So, what is biological problem? Biological problem is that we don't know till now the real diversity of 4 repeat channels because a part of metazoa and fungi, there are more than 20 taxonomically equal groups of eukaryotes. Yeah? The second reason is fungi and animals are very close relatives. So, in eukaryotic point of view, it's almost the same point. So, we found 277 amino acid sequences from metazoa, fungi, and 16 other eukaryotic groups, and make some phylogeny with maximum likelihood and bias analysis. This is what we inferred. That 5 subfamilies are in red, and subfamilies of metazoa and fungi, and is a group of close relatives here. What we show that most eukaryotes possess their own subfamilies of 4 repeat channels, which not clusterize with any known subfamily. I mean, not with calcium subfamily or sodium subfamily. So they possess their own 4 repeat channel. I mean that when you read in some papers about voltage-gated calcium channels of paramecium or of green algae, it doesn't mean that it's, for example, A-type channel. It's absolutely another calcium channel, just from parallel evolution. Okay, and the second thing is that the diversity of subfamilies in many eukaryotes, and most of eukaryotes are unicellular organisms, yes, are comparable to that in in animals, yeah? I mean, human possess 4 subfamilies, and for example, dinoflagellates possess at least 4, and ciliates possess at least 6. Okay, so often phylogenetic diversity somehow reflects functional diversity, yeah? And so we decided to analyze the primary structure in some important lots of these channels. First of all, is so-called selectivity filter motif, and it's well known that 4-glutamate is responsible for calcium selectivity or permeability, and even DEEA from voltage-gated sodium channels are to calcium, and the famous DECA or EEKE means sodium channels at least sodium permeable. So we can not judge about the degree of selectivity, but we can expect that the channels will be sodium permeable. Okay, so this is just an interesting example, but what we found? We found that most of channels of eukaryotes, most of 277 channels possess 4-glutamate selectivity filter, and this confirms the old theory about from Ilya and Armstrong about that ancestral channel was calcium. It's look like, yeah. And this is an interesting example. It's a huge clade of 4p channels of ciliates, and this clade can be divided in two subgroups. Group which possess canonical 4-glutamate selectivity filter, and unusual for aspartate. So here I can expect that this channel will be calcium, and indeed we know about voltage-gated calcium channel in ciliates, but I can even imagine about selectivity in this subgroup. Okay, the most interesting point was to find maybe sodium selectivity in unusual groups of organisms, and indeed, as I mentioned, the sodium selectivity is somehow related to lysine to K in second or third domain. So we found these channels with such filters, for example DDKD, and very close to DECA AGKE in oomacetes. Oomacetes is parasites of animals and plants, and these organisms are filamentous. And every neurobiologist knows that in filamentous structure it's a big advantage to split sodium and calcium signaling, so at least it have a sense. And another interesting example that we found is not lysine but arginine. So it's interesting to answer influence of this residue on selectivity. Second locus was segment S4, so-called voltage sensor. It's well known that when this sensor regularly composed from arginine and lysine, these channels are voltage sensitive. But for example, in sodium leak channels or calcium channels of fungi, there are segments S4 that are poor with positive charges. So this example of dinoflagellate channels, and there are a lot of channels with a normal S4 segments, and some channels with a poor with segments that poor with positive charges. So we can suppose maybe these channels are voltage insensitive. And again, the examples is again ciliates. So like in human, ciliate possess the whole clade of channels that I suggest to be voltage insensitive. And for example, dinoflagellates possess a huge clade of channels, and this clade is some mixture which made from channels that are supposed to be voltage sensitive and channels that are supposed to be voltage insensitive. Third interesting lot is so-called inactivation gates, but not simple inactivation gates. I mean inactivation gates of that characteristic to voltage-gated sodium channels. The famous EFMT motif, three hydrophobic residue and one polar residue. So 3 green, 1 blue. This example is LVL channels, and everybody knows that LVL channels didn't possess that structure. Indeed, we have or deletion or substitution to positively or negatively charged residues, is red squares. So they don't possess fast activation like sodium channel. But we were surprised when we found that the common situation that most of eukaryotic channels possess this structure. Structure that we thought before characteristic only to sodium channels. No, it's a predominant structure, and that's why I suppose that ancestor state possess the inactivation gates, but it doesn't mean that this channel, the channel have a fast inactivation. Yes, it just had a structure. So then we switch. First interesting motif is IQ motif that's responsible to interaction with calmodulin. Again, the canonical structure of calmodulin, I mean IQ or LQ, 3 something, are okay, 4 something, and then again are okay. We found this structure, of course, in sodium and calcium channel. It's well known, but in the same time we found the structure in very different, not related to each other groups of eukaryotes. So again, we suggest that this state was in ancestral channel. But in the same time, there is some diversity. What I mean, for example, the same organism can possess the channels with a canonical IQ motif and with somehow changed IQ motif, or even with reduced, I mean deletion, just deletion. It should be secondary deletion. But we found linear specific. So we confirm linear specific gain that only A type of calcium channels have a second IQ motif in the chain. So, and the last interesting motif was the link between repeat 1 and 2, which contains the so-called alpha interaction domain, the domain which is needed to interaction with beta subunit of calcium channels, of HVA calcium channels. And I would like to say that it is very abundant, but I can't. So it's a linear specific gain. So only HVA calcium channels possess a subunit for interaction with beta subunit of calcium channels. So, okay. Then we can imagine what did the ancestor of calcium and sodium channels look like. I mean, what did the ancestor of all of 4 repeat channels look like? First of all, we think that it was voltage sensitive. It's interesting because today we believe that the 4 repeat channels evolved from some TRP channel. So, and TRP channels are voltage sensitive. Second, it's we think it could be calcium channel because the 4-glutamate state is most abundant, and we found it in very diverse groups of organisms. Third is it possesses an activation gate motif, but we don't know, of course, about the fast inactivation itself. And then it was able to attach calmodulin, but wasn't able to attach beta subunit of calcium channel. So that's all. Thank you.
Thank you. This paper is now open for discussion. Yes, please.
I have a comment on the side. For example, 4-glutamates in the classical selectivity filter locus does not necessarily correspond to the calcium selectivity because, so everybody, it's well known that there are bacterial sodium channels which are symmetric tetrameric channels, and they have glutamate in this position, so we have 4-glutamate in the ring, and basically
nobody knows
sodium selectivity. So just can recommend to be more, say, careful because functional properties are not so simply linked to the sequence elements.
Yes, it's of course. So it's only our prediction, our expectation. Then we need to produce some real experiments. But there is only, till now, there is only one exception in 4 repeat channels. I mean, in terms of 4-glutamate selectivity filter. Only a part of this exception, all of known and measured 4 repeat channels are calcium. So it's a phenomenology just. So
yeah. It can mean that there is another determinant structure besides the symmetrical glutamate link. By the way, one of the possibilities is what, say, to the best of my memory, calcium channels in the second repeat contain very conserved aspartate just downstream from the glutamate ring. And to the best of my memory, this aspartate is very essential to for the calcium selectivity.
Yes, it's just because the selectivity is determined not only from this, it's only size of the most strains. How to say? I mean, the selectivity is arise from the whole P-loop, of course. So it's just first step.
I actually have a question. Oh, yes, go ahead. Mike.
In evolution, deletion events have been described as very important. So if we assume that a 4 repeat channel, whether it's calcium or sodium, I don't care at this moment, but that this is an ancestral, how do you then explain that the abundance of monomeric, let's say, repeat channels, KV channels, strip channels, whatever, is, let's say, the next finding? So where are the trimers, where are the dimers?
So dimers exist, so they are abundant. For example, in our lysosomes, and even the gamolgas exist in the plants. In what I believe that the last common ancestor of all eukaryotes have a very fast evolution, very fast duplications events. So in the ancestral state, and now we have a heritage of these events, and why monomeric channels exist, just because do you know that many eukaryotes have not like animals are bad eukaryotes indeed. So, but many eukaryotes possess a lot of copies of the same gene. So of course, every copy can evolve how it evolved. Yes. So I think maybe the last common ancestor have something like this, and so we have a whole set. But I don't know, of course, about trimeric channels.
Thank you, Ilya, once again.